Yeah. So Niki currently is senior investigator, also chief of oral immunity and infection section in National Institute of Dental and Craniofacial Research Institute, NIH. Very, very brief introduction. Niki got her Doctor of Dental Surgery degree from Aristotle University in Greece. And then after which she completed her specialization in periodontology at the University of Maryland and obtained her PhD in immunology at NIDCR. So Niki has been working as a clinical fellow at NIH, specifically NIID with Dr. Steve Holland. And since 2010, Niki has been an independent investigator building a clinical and translational program, which I think we're going to hear a lot of translational work today, focus on basic oral immunology and then susceptibility to the common human inflammatory disease, periodontist, through the study of patients with monogenetic immune defects. So Niki's research is mainly focused on oral barrier immunity, implement a bedside to bench approach and aim to understand the molecular and cellular basis of oral immunity in health and in common inflammatory disease of periodontist. And then she's really interested in studying the oral inflammation, dysbiosis, which is also associated with infections in other parts of the body, as well as systemic inflammatory conditions. Her lab mainly aims to understand the machinery which microbiome triggers and host elements used to regulate oral immunity in health and disease. So actually, Niki really made a lot of groundbreaking discovery regarding to the oral barrier immunity, which I think all the mucosal immunologists learned a lot. With these, she got numerous awards, which including the Distinguished Female Mentor Awards, and then also Harry Curry Society elected member. She also got last year actually, Distinguished Scientist Award for facial biology research in oral biology. I think as a mentor, she's really raised a lot of outstanding trainees who are currently very active in both academia and industry, which I'm super impressive. So without further ado, the floor is yours. Please go ahead.
Thank you, Sean. That was a very generous introduction. And that's what I'm planning to discuss today, give a little bit of an idea of how we've been studying oral mucosal immunology through the lens of Mendelian immunodeficiency. And these are studies that really can uniquely be done at the NIH. So I'm going to only highlight a couple of them, but I think it will give everybody an idea of the kind of work that we're doing. And so as Sean already alluded to, we're interested in barrier immunity and barrier sites of the mammalian host, such as the skin, the gastrointestinal tract, the urogenital tracts, the respiratory areas are all exposed to the outside environment. They're exposed to elements of the air, of food, of a microbiota that are commensals or pathogens. And the systems of the immune system at these barrier sites has to be able to tolerate commensals and innocuous antigens, but at the same time protect us from pathogens. And so the fundamental questions that apply to all barrier sites are sort of common, such as how is tissue integrity maintained? How are we protected from pathogens? How are we tolerating what is commensal and innocuous? But at the same time, with work that's been done over the last 2-3 decades, it becomes very evident that a lot of the answers are tissue specific. So while the questions are shared, the way each body site is able to control and these regulatory mechanisms becomes different. And part of that has to do with the fact that every tissue has its unique property and functionality, but also its unique exposure. So in the gastrointestinal tract, we get exposed to the food, but in skin, you get exposed to sunlight. And so each environment has to mediate those those mechanisms in a different way. And so it becomes then very important to study immunity at a specific site. And so again, as Juan already mentioned, we are very much focused on oral barrier immunity. I and many of my fellows in the lab throughout the years are clinically trained as dentists. And so we have an interest related to the diseases of the mouth. But for those of you, and I'm assuming it's the majority of you that are basic scientists or physicians, the oral cavity also becomes an area that is very understudied. It's an area of the human body that is exposed to the outside environment and to many of these exposures for the first time. So, for example, the commensal microbiota that go into the gastrointestinal tract will start from the mouth. Food will start its journey in the mouth. A lot of the airborne allergens and particles will be breathing in both from the nose and the mouth. And so we have a lot of exposures that are first encounters here at this body site, and we don't understand the biology of it that well. We don't understand either how the system is mediating tolerance or how imbalance between the outside and the host leads to disease. And so we're interested in both from the standpoint of understanding local immunity, but also because a lot of these elements are encountered for the first time here, with the idea that these first encounters could be playing a role also in modulating systemic immune responses. And so from the standpoint of disease, though, I also wanted to introduce here our disease of interest, which is periodontitis. And this, again, Xuan already mentioned, this is a disease that we're working on. It's a disease of the oral cavity. It's extremely common, seen in severe forms in about 8% of the general population. This is a little bit of a diagram of a cartoon of how we think about it. The teeth sit within the alveolar bone. And the bone is covered by mucosa. In health, there is a homeostatic and health-associated microbiome, but the host is able to tolerate it and not lead to inflammation. However, in the context of disease, we have a dysbiotic microbial community, an inflammatory response in the mucosal tissues, and this mucosal inflammatory response becomes pathogenic and drives bone loss. The interesting thing here is that you may have inflammation on and off, and many of us do in the mouth. However, it doesn't always drive destructive periodontitis in all individuals and not to the same degree. And so this is a disease of interest. It's associated with systemic other diseases. So as a disease itself is interesting, but also it becomes an area where we can start understanding host microbiome immunity. And it lends itself to understanding host microbiome immunity in humans because it's easy to access the oral cavity, to see disease and inflammation, to harvest microbiota, to harvest tissues. And so that's allowed us to do a lot of clinical and human studies over the year, not only in our lab, but of course, a lot of other colleagues as well are doing this. And so, but what we're doing that is really unique, and that has to do with the environment of the NIH Center, is the study of Mendelian diseases, where we see patients that have single gene manifestations or single gene disruptions, and these diseases then are associated with a clinical phenotype, which is often severe. And these types of patients are important for us to treat because they're rare. And so we can see them at the NIH all together and help them because many of them are having a hard time having care outside. But at the same time, they become, in a way, models towards us understanding the role of specific genes in immunity. And in our case, specific genes in the susceptibility to periodontal disease. And so we see a lot of these Mendelian disease patients. We characterize the ones that have oral manifestations, and then we take a lot of samples in the lab from human subjects, which we work up, but at the same time also try to establish animal models in order to understand mechanisms of disease and also, when possible, treat it. With the idea that ideally we want to understand rare disease to eventually understand subtypes of common forms of disease. And so I'll give you one example today, which is one of the kind of studies that have, in a way, gone the full spectrum from Mendelian disease to mechanism and to giving us a beginning of understanding potentially for systemic diseases. But I will also present kind of some of the efforts that we've done to create tools towards the study of human oral immunity. So I'll start with the tool part. I'll say that both in the mouse and in humans, the types of assays that were present were not many. People have not been working on tissue immunity, especially in humans, much. And in the oral mucosa, even less. So we've done a lot of work over the last decade to take tissues and do a lot of not only histological analysis, but also dissociation of tissues, flow cytometry, understanding of immune cell populations. But more recently, and as single cell sequencing became a lot more in fashion, for lack of a better word, we made this big effort of trying to build a human single cell transcriptomic atlas of oral mucosal tissues. And for this work, we bring patients and healthy volunteers within the NIH in our clinic. We run both a clinical program and a basic lab. And the work was spearheaded by clinician scientist Drake Williams, which, as Xuan said, I'm part of my passion in life is to try to train the next generation of clinician scientists. And Drake last Monday started his own lab in Minnesota, has a K99, so I'm very proud of him. So when Drake was in the lab, we compiled the human oral mucosal atlas. We brought in healthy volunteers and we screened them very carefully and took tissues both from the lining mucosa, which is in the inside of the cheek, and the tooth-associated mucosa, which is next to the dentition, otherwise called gingiva, with the idea of getting representative sites of the oral mucosa and understanding both this local area that can lead to periodontitis, as well as the more general area of the oral cavity. And so the pipeline that we used is not only using single cell, but also trying to confirm by flow cytometry and histology some of the things that we're finding. This gave us the opportunity of starting to look and confirm the presence of basic cell populations that we knew were there, epithelial, endothelial, fibroblast, and immune cell types. But also try to look at the subtypes within them. And I won't take you to great lengths. Oops, sorry, I lost something here. I won't take you to the great lengths of the types of cell populations that we found, but you'll see here, even in health within the major immune cell subsets, we have a big representation of myeloid and the granulocytes. T and B cells really are the biggest proportion in health and very small proportion of B and plasma cells in the setting of health. But what became even more interesting to us was the fact that we started to characterize non-immune cell populations. And what we found here, which was very interesting and really unexpected, is that both epithelial cells, endothelial and fibroblasts, had specific subpopulations, but in all of them, there were specific subsets that had inflammatory profiles. So, for example, we find a subset of epithelial cells that have a lot of potential for neutrophil recruitment, at least theoretically. We find different types of endothelial cells that have immune kind of associated transcriptomes, anywhere from antigen presentation to chemo attraction. And within the fibroblasts, again, we see subtypes of fibroblasts that are either associated, again, with neutrophil recruitment or with tissue turnover and complement activation. And here, between gray and yellow, I'm showing the proportions of these cells within the different parts of the oral mucosa. And we see that a lot of these immune active cell types are mostly within the gingiva, the tooth associated, especially those that have to do with neutrophil chemo attraction, and that becomes of interest later. We've also built a portal that's very nicely interactive together with some colleagues in Newcastle University in the UK, where people can go and mine our data in a kind of very easy and straightforward way. And so we've been building a lot of these atlases and tools, both for ourselves and for the community, but part of what both in health, but also in periodontitis, which I'll show you here. And in the setting of periodontitis, we take biopsies from health and periodontitis to compare. You'll see here in histology the level of inflammation within the periodontitis tissues. We know by flow, that there's tremendous accumulation of CD45 cells. We see the same within the immune cell subsets of the single cell. And I won't take you for, in the interest of time, to cell type per cell type within perio and health. But what I'll say is that one interesting observation that came out of our atlas is the fact that these non-immune cells, both epithelial cells, but even more intriguingly, stromal fibroblasts, seem to have transcriptomes that are consistent with immune cell activation, and particularly with granulocyte recruitment. These are present in health and very specific to the gingival part of the oral mucosa, and seem to very much become activated and expansive in the setting of disease. And so this has also opened a number of studies within our lab thinking about the role of non-immune cells in the setting of periodontitis. I'm going to stop for 1 second because I lost the image of myself and others. Xuan, can you see me? Okay, sorry, I will go back up.
Well, I guess you're fine.
Yeah, I just lost the part where I could see me and you, and then I feel like I'm in the air talking, which is very odd. Here's this one I want to share. Can you see my screen now? No.
It's
Stop
not
sharing.
on the play mode.
It is not on the play mode. How do I do that?
On the bottom, you have the play mode, just the full screen, full screen play, the PowerPoint. What, like exactly what you
No,
did before?
wait. I don't see the PowerPoint though. Do you see my PowerPoint?
Yes, I do.
Oh, now I see it. Okay. I don't see me, but it's okay. I was hoping to see our faces at the same time. So I'm going to switch gears now, as I mentioned, to kind of present in the last 20, 25 minutes, a study that I am particularly excited, although it's not very recent, but it's really one of these studies that has allowed us to go from rare disease to starting to think of how that could apply to common forms of periodontitis. And this is a disease called plasminogen deficiency. It's a rare Mendelian disease caused by mutations in the plasminogen gene. And plasminogen, mutations in this gene lead to reduced plasminogen enzymatic activity. This is an enzyme. And patients that have these deficiencies do present with a wide spread of mucosal immunopathologies. And so they have ligneous conjunctivitis, they have these mucosal lesions in the eye, and periodontitis, and really this is the most common manifestation seen in about 80% of the patients, but also a 20 to 50% subset, depending on the phenotype, will also have gastrointestinal immunopathology, lung immunopathology, and vaginal immunopathology. And so as we start to kind of give a little bit of a background on what plasminogen and plasmin are, is plasminogen is the inactive form of the enzyme plasmin. When it becomes activated, it becomes plasmin. And the main role of plasmin, and this, of course, you know from basic biology, is to break down fibrin clots. So when you have an injury to a vessel, when you have inflammation, you will have bacteria, you will have leakage of vessels, and you start formation of a fibrin clot. And these fibrin clots are very important in order to achieve hemostasis. However, they also need to go away. They can't stay there forever. You need an enzyme that will degrade them for wound healing to occur. And so if, however, plasmin, who is the main enzyme for that, is not there, then you have tremendous fibrin accumulation, and that does not allow for wound healing and can cause inflammation. And really, that is what people have shown in the tissues of patients with plasminogen deficiency. There's these tremendous fibrin deposits in mucosal tissues and later on in life throughout the body. And so, of the fibrin not being cleared. And so, this is an interesting disease But what made us able to start to study it was the fact that Thomas Buga, who's a colleague at NIDCR, many years ago had made the plasminogen knockout mouse. And this mouse very closely phenocopies human disease. So spontaneously, these mice will have the ligneous conjunctivitis, will have a shortened colon and anal prolapses within weeks, a systemic inflammation, both and regional inflammation, as well as what we now show, also ligneous conjunctivitis. And so Thomas came to me with this idea of collaborating, and so we recruited this fellow to both of our labs, Lakmali Silva. Lakmali also is an independent investigator at Harvard Dental School, and she started her lab a couple of years ago. And so Lakmali came and started working on the plasminogen knockout mouse, and with asking the question, how does plasminogen deficiency lead to mucosal immunopathology? And so here I'm showing you a micro CT scan of a knockout versus a wild type mouse. And what you can appreciate is that the bone levels are much further away from the tooth in these plasminogen knockouts. And that is kind of a feature of periodontitis. This bone loss is spontaneous. And so when she looked at the knockouts versus their littermate controls, this bone loss is significant by 11 to 12 weeks of age. And by 23 to 24 weeks of age, this becomes tremendously severe. And so we have a spontaneous phenotype here. And so we can say that our animal models phenocopy human disease for the oral phenotype as well. And so when Lakmani looked at the mucosal tissues, and this is the soft tissues around the tooth, what you'll see here is these accumulations of fibrin within the tissues, similar to what people have seen in patients. And this allowed us to ask the question, is the fibrin what drives immunopathology? And so to answer that question, we did a simple kind of experiment, of a genetic experiment. Lakmali crossed the plasminogen knockout to the fibrinogen knockout mouse and asked the question of, if you take fibrin out of the equation, is the disease going to be rescued? And in fact, we saw a complete rescue. And so here you'll see again the severe phenotype in the plasminogen deficiency, and that it's rescued when you don't have fibrin. And you can also see here with all the different crosses that the disease is rescued almost to a better degree than what the wild type control has. And so that then answered the question of, yes, fibrin is the driving force of immunopathology, and opened up two different questions of, why is fibrin triggered here in the mucosal tissues, and what are the mechanisms by which it causes immunopathology? And so what we did first for the trigger part is that we assumed that if you have a phenotype mostly at mucosal sites, it's likely that the microbiome, which is across the mucosa, and would play a role. So we rederived the mice germ-free. And what we saw here with the different measurements that you'll see is that really the germ-free mice are significantly protected. They were not 100% protected, but they were significantly protected. And you can appreciate here kind of the swelling, the accumulation of fibrin, the loss of bone that we see in the SPF mice, as opposed to the germ-free mice, which have nice bone levels, nice collagen tissue, lack of swelling, lack of this accumulation of fibrin, and we quantified all of these measures as well. And so then that gave us kind of at least a partial answer that the microbiome is super important here. That kind of explains many of the mucosal phenotypes, and it drove us to focus a lot more into the mechanisms by which fibrin would trigger disease. And so we first took tissues around the dentition of the mice and did kind of a non-bias approach of RNA-seq bulk sequencing. And we saw the differential pathways that came up were mostly immune associated, with neutrophil-related pathways being on the top of this list. And in fact, when we looked at dissociating tissues and doing a flow cytometry within these tissues, neutrophil accumulation was the top kind of characteristic. So we looked at this at 12 weeks of age, which is the beginning of the significant bone loss. And at that point, neutrophils were the only cell type that was significantly associated. You'll appreciate it here by Lysis G and CD11B staining, how much more it is in the knockout versus the wild type. What we saw is that at later time points, myeloid cells, macrophages also accumulate. However, at early time points, the only cell type that was significantly upregulated, at least of major cell types, were the neutrophils. And so we also saw this at the tissue level, and for me, histology is always a lot more beautiful. We see now with specific fibrinogen staining, the presence of fibrin in the knockout, and Lakmali has a very nice control here using the fibrin knockout mouse. And we see also the accumulation of neutrophils within the environment on top of the fibrin, around it, and really everywhere. And so then our question becomes, could there be an association or interaction between fibrin and neutrophils that drives this problem? And the reason this made sense right away to us, or that we really had a bias towards this, was the fact that we already knew that fibrin has a domain that is a binding site for CD11B found on all myeloid cells. And interestingly, this domain is a cryptic site. So when fibrin is in the blood circulation, this is not shown. It's only when fibrin is cross-linked within a clot that this opens up and myeloid cells can bind it. And the other very fortunate thing is that a collaborator of ours, Matt Flick from UNC, had made a mouse that was specifically mutated on fibrin in the site of binding CD11B. These mice have normal amounts of fibrin. The fibrins can form clots. They have no bleeding issue, but they just cannot bind myeloid cells. And so then Lakmali did this very nice experiment saying, if I cross plasminogen knockout mouse to this fibrin specific knockout, where you still have fibrin, just not the binding of myeloid cells, can I rescue the phenotype just by taking away the binding of myeloid cells? And this, in fact, was a very exciting result. It was beyond our wildest dreams. So the knockout, again, you see, has significant bone loss compared to the wild type. However, when we use this Fib gamma knockout cross to plasminogen, we have significant protection actually to a better level than the wild type itself, indicating that this binding of myeloid cells is key. And in fact, what was also interesting is that in the tissues of these cells, we didn't have reduction of fibrinogen. We also did not, and I'm not sure, I don't have this, but we also don't have reduction in the number of neutrophils. And that was really interesting because in general, we think of neutrophil accumulation as that equating to immunopathology. And what we're saying here through this work is you really need neutrophils to be bound and activated potentially for immunopathology to occur. If you have tons of fibrin and tons of neutrophils together without the binding, it doesn't cause a problem. And so then Lakmali went on to create an in vitro system where she could look at what happens when neutrophils bind fibrin. And what she did is she created these plates where she would lie down and cross-link fibrin, either from wild type or knockout mice. And so she would use either human or mouse neutrophils for this and say, what happens if I add neutrophils here? And right away, the first thing that she saw is that on the Fib gamma fibrin, which is mutated, she has less attachment. So to begin with, these neutrophils cannot adhere. And she also did the same with the opposite kind of experiment where she used wild-type fibrin, but took out CD11B from the neutrophils. And again, attachment is reduced. She also did a number of functional assays where she said, if I add neutrophils into this fibrin, and I add very low levels of PMA, not enough not to create a severe activation. How does ROS production, MPO production, degranulation, netosis look when these neutrophils are attached to fibrin versus not attached when the Fib gamma is there? And so she saw significant reduction in ROS production. She also saw significant reduction in NETosis. And in fact, this for me is also kind of a very nice image to show. She did a lot of this live imaging where she looks at over a number of hours at NETosis after adding low levels of PMA. On the left-hand side, what you will see is when neutrophils are in wild-type fibrin versus knock-in fibrin. And I think you'll appreciate, you'll see the neutrophils blowing up. They become green when they're permealized and red when they pretty much explode into a net. And you'll see these kind of little bombs coming up on the wild type a lot more than in the knock-in. And this is represented also by the quantification that I showed you before, but it's much nicer, I think, as a video. And so what Lakmali was able to show is that the adhesion of neutrophils on fibrin through Fib gamma allows for their activation if they get the right stimulus. And this activation within tissue, we suspect, plays a role in immunopathology. And so she then asked the question whether if you inhibit specifically NETs, for example, within this model, would that lead to reduced immunopathology? And in fact, she did a number of ways of doing this. She either treated mice with DNase or used specific genetic knockouts. And what she saw is that if you remove NETs by DNase, you can protect mice. It's not as significant of a protection or complete the way Fib gamma was, but it really is part of the story of trying to, of how we are led to immunopathology. And here are also the genetic models that she's used to double and triple confirm her findings. And so we were excited about this, not only for the sake of periodontitis, but we think that we have uncovered here a mechanism that could be globally important for wound healing. And so, for example, we know that in the setting of a wound, you want fibrin to accumulate and create a clot. And you want that clot to be able to bring in myeloid cells, especially neutrophils, because you want the neutrophils there around the clot to be able to perform all the antimicrobial functions such as ROS production and NETosis. You want them also activated efficiently in that setting so they can do their job. And then eventually when the wound is cleared, you want these neutrophils to also be able to phagocytose and fibrin to go away. And by plasmin and all of the wound healing to occur. And so we think that this binding of fibrin to neutrophils is homeostatically an important wound healing mechanism. However, if fibrin keeps being there, either because there's continuous stimulation or because there's a defect in plasmin and plasminogen, then this, which was supposed to be a protective mechanism, becomes a pro-inflammatory mechanism that does not allow for wound healing. And so we're interested in this from the setting of Mendelian disease, from the setting of biology, but then we took it one step further and said, could this apply to any of the common forms of periodontitis? And so we were very fortunate to collaborate. Before I tell you that, I will tell you that it made sense to us that this could be relevant for periodontitis in general, because periodontitis is a disease where there's abundance of neutrophils and abundance of fibrin and bleeding. And so here, these are neutrophils within the tissues of periodontitis patients, but these are also stainings where you'll see fibrin deposits and MPO staining of neutrophils within similar areas in periodontitis. But we were also fortunate that around the time that we were doing this work, a paper came out from Germany showing that it was a big GWAS study in aggressive forms of periodontitis. And that paper showed that one of the top genetic traits associated with aggressive forms of periodontitis was actually a hit in around the plasminogen gene. And so that kind of told us that this could be relevant to common forms of periodontitis, but we also did a secondary study collaborating with Kimon Divaris at UNC, who had a smaller cohort. And what he saw within the US cohort, again, was that severe periodontitis is associated with these genetic traits around the plasminogen locus. And this is also associated with a carriage of specific microbe called Actinomyces aggregatibacter actinomycetin commentans, which is associated with aggressive forms of periodontitis. And so what this is now telling us is that this allows us to start thinking of the fact that Mendelian diseases such as plasminogen deficiency might help us characterize specific subsets of periodontitis in the common population. Obviously, we have a long way to go in understanding what those polymorphisms mean in the general population, but it's giving us this idea that we want to look at subsets of these patients based on the polymorphism and see what the functional role of plasminogen polymorphisms is in the general population. And so I just want to leave you with this idea of, in general, the type of work that my lab and clinical program have been trying to do at the NIH is to create kind of a baseline of assays and ways of looking at oral mucosal immunity in mice and humans. And what we want to eventually do is start with Mendelian diseases, do studies in the lab with tissues and cells of patients. When possible, phenocopy human disease, understand mechanisms, perform preclinical studies with the idea of leading to human interventions for Mendelian disease, but also potentially expand this work and observations for Mendelian disease into traits that could be associated with common forms of periodontitis. And so I leave you with this message and maybe with the most important slide with acknowledgements. I already mentioned Drake and Lakmali that did the majority of the work in the studies presented. These are other members of the lab. And obviously this work, especially the clinical translational work, needs a lot of partners in order to be accomplished. One of our major partners are Steve Holland and Mikhail Zvionakis on the clinical end, as well as from the basic science within the NIH, Yasmin, Giorgio, and in the extramural, George Kajicengalis and Joe Conkle, among many other colleagues. And I'll stop sharing and happy to take other questions.